Transcription is the synthesis of a complementary strand of RNA from a DNA template. Let's think of transcription as making a new bracelet based off of an old one. Here is my two-stranded bracelet. I'm going to make another bracelet for my friend, but she only wants a single-stranded bracelet, and she wants one that doesn't have the color pink in it. Let's look at how this could occur biologically. RNA polymerase is an enzyme that builds an RNA strand, or new bracelet. Let's think of RNA polymerase as our master bracelet builder, who reads our original bracelet pattern and picks out what beads should be put on the new bracelet. The core enzyme of RNA polymerase is composed of beta, beta prime, and two alpha subunits. When combined with the sigma factor subunit, these components come together to form the entire holoenzyme, which is important for the initiation of RNA or bracelet synthesis. RNA polymerase first binds the DNA, or old bracelet, at the promoter region, a part of the DNA sequence that is vital to the regulation of the expression of a gene. Let's think of the promoter region as a special code that shows our master bracelet builder where and when to start building the bracelet before they actually start building it. Two vital sequences in the promoter region are the -10 box and -35 region. They are important DNA promoter sequences that specify to other molecules where transcription begins, ensuring that RNA polymerase will bind in an appropriate spot of the DNA. The sigma factor of the holoenzyme is what recognizes the -10 and or the -35 region of the promoter and tells RNA polymerase where to bind. Without the sigma factor, RNA polymerase would not be able to find its binding site and transcription could not occur. The binding of sigma factor signals DNA strands, or our old bracelet strands, to separate, forming a transcription bubble. This allows us to get a better look at what we want to build for our friend. RNA polymerase core enzymes start synthesizing our bracelet at the +1 site of our DNA, or old bracelet. The +1 site is important as it signals the RNA polymerase to add the first nucleotide, or bead, transcribed into RNA. RNTPs are the building blocks of RNA synthesis. Think of them as our bracelet beads. RNA polymerase reads the template DNA strand in the 3' to 5' direction. It adds complementary RNTPs according to the base pairing rules of A and U and C and G onto the 3' OH group of the previous nucleotide. It synthesizes the RNA strand in the 5' to 3' direction. RNA polymerase catalyzes the formation of important phosphodiester bonds, or our string, that are what ultimately join the nucleotides together into a strong strand. The coding strand of DNA is what the produced RNA or bracelet will be identical to, other than the replacement of T with U, or pink with blue. Termination hairpins are important to the processes of ending transcription. Row-independent termination involves hairpins that are followed by a string of adenine and uracil subunits. These are weak segments causing RNA polymerase to fall off and transcription to end. Row-dependent termination occurs when a row protein binds to the RNA being formed. A termination hairpin causes RNA polymerase to pause and row catches up to it, forcing it off and ending transcription. Transcription is a complex process. If only one thing goes wrong, the cell may not be able to function. For example, if the -10 box is deleted in a gene, then the sigma factor of RNA polymerase can't properly bind to the DNA sequence and transcription from DNA into mRNA would not occur. If this gene encoded an enzyme to metabolize sugar, for example, then the mRNA needed to build the protein enzyme would not be present. The protein enzyme could not be made, meaning the cell would not be able to metabolize sugar.